We've now discussed a lot of different smaller units on the organization of life. We've seen atoms, we've shown how atoms can form molecules, we've seen how those molecules can interact with each other. We've also seen how atoms and molecules can form macromolecules, and the effects of interactions between macromolecules can help maintain homeostatic processes in living things. Now let's move on into the next larger group in the organization of life. A cell is the smallest unit of life that can function independently. A cell is very small. So how can we see such small things? This scale presents different objects in different magnitudes of size. To the left on this scale are objects that are large and can be measured using meters. On the far right are things that are so small that they're best measured using nanometers. The human eye can see things about as small as 100 micrometers. This is about the thickness of a human hair. A light microscope can see things that are smaller, down to about 100 nanometers. To see things smaller than that, you need a more powerful microscope, something like an electron microscope. Hopefully, using our light microscope, we'll be able to see things as small as a single cell. Let's look at the anatomy of a eukaryotic cell. Eukaryotic cells have ribosomes, structures that manufacture proteins. They also contain DNA, which is genetic information. Cells also have a cytoplasm, which is a fluid-filled portion. Lastly, cells have some kind of cell membrane. In this case, it's a lipid-rich layer that separates the internal and external environment. Other living things, like fungi and plants, have other layers like cell walls that provide an additional layer of protection. The cell membrane is made of a phospholipid bilayer. On the far left is a single unit of the phospholipid bilayer. This is essentially one molecule of a lipid. On one side is a hydrophilic head. It's called a hydrophilic head because it has an attraction to water. The other end of the phospholipid, which includes the fatty acid tails, is hydrophobic. It repels water. An interesting characteristic of hydrophobic molecules is that hydrophobic molecules attract one another. So, if you put a lot of different phospholipids in a solution, they tend to organize themselves. The hydrophobic tails attract one another, while the hydrophobic heads tend to orient themselves towards the water. To the right, you can see how this automatic organization looks. You can see that the hydrophilic heads have oriented themselves outwards, touching the water. This is because they're attracted to the water. The hydrophobic fatty acid tails have repelled away from the water, but they're attracted to one another. The end result is that the phospholipids form two layers. This phospholipid bilayer forms the outer membrane of a cell. Embedded in that layer can be a lot of different other structures. Here you can see there's protein channels, there's hormones traveling through, there's filaments of the cytoskeleton below to maintain its structure. There's also membrane proteins. All of these different structures are embedded within a matrix of phospholipids, and these phospholipids maintain their shape due to forces that don't require energy input from the cell. Plant cells also have cell walls outside the plasma membrane. These cell walls help maintain the cell's structure. As the cells build pressure, those cells become rigid. The chemistry of the membrane on the outside of a cell can help us determine what domain the cell belongs to. Bacteria have a fatty acid membrane. They also don't contain organelles and don't have a nucleus. Archaea, which are similar to bacteria in many respects, have a non-fatty acid lipid membrane. They also do not have organelles and do not have a nucleus. Eukaryotic cells have a fatty acid membrane. They also have a nucleus and organelles. Let's explore the different organelles in eukaryotic cells in cellular anatomy. Eukaryotic cells have a nucleus. The nucleus contains DNA. It also has a nuclear pore and a nuclear envelope. The nucleus contains much of the information that the cell uses to manufacture different proteins and protein products throughout the body. The endoplasmic reticulum lies outside of the nucleus. The endoplasmic reticulum contains a concentration of ribosomes. Those ribosomes build proteins. The Golgi apparatus further folds and builds proteins and adds identifying carbohydrates to molecules. The Golgi apparatus also contains vesicles. Vesicles help transport products in and out of the cell. Lysomes are organelles that contain enzymes. These enzymes can break down products. Lysomes are much like a stomach and waste structure in the cell. The enzymes inside lysomes tend to function at high pHs. Mitochondria perform cellular respiration and provide much of the energy in our bodies. Chloroplasts help produce much of the energy in autotrophic organisms through photosynthesis. Another feature of cellular anatomy is the presence of things like flagellum or cilia. Flagellum and cilia are used in locomotion. On the bottom left, you can see an example of a euglena. This is a unicellular organism, a protist. On the left side, you can see a whip-like appendage. This is a flagellum. As the flagellum moves about, it creates a propeller-like motion. On the far right, you can see an example of a rotifer. This is another unicellular organism. On the top right of that rotifer, you can see kind of a fuzzy area on two pads. Those areas contain many small hair-like features that beat back and forth much like a flagella.